Hi, welcome back to part 3 of chapter 12 lecture video. And this chapter is not as long as the muscle chapter. This part 3 won't take another hour. It would be pretty, maybe 30 minutes or so. So for one last time, I will go over the steps using the animations I've drawn myself to for sure that you're very clear what's going on in the action potential. So what happens is, in the resting membrane potential, you have lots of sodium ions on the outside and lots of potassium ions on the inside. The cytosol is negative charge, whereas outside of the membrane is positive charge. And we're currently at negative 70 millivolts. At negative 70 millivolts, both channels are closed. You have a closed sodium ion gated channel and a closed potassium gated channel. And know that these are your voltage gated channels. So right now we are currently at this position of the depolarization action potential graph. So when we have a depolarization towards threshold right here, so we're talking about this climb in membrane potential, what happens is your stimulus comes through and enough greater potential has generated to your membrane to increase your membrane potential to be negative 60 millivolts, which activates the opening of your sodium gated channel, allowing sodium ions to flood into the cell, into the neuron. And remember that this is an all or none principle, meaning that once that action potential is fired, there's no going back. We can't just say, hey, let's stop in the middle of the neuron and then not get to the axon terminal. That doesn't happen. Once it's fired, that electrical signal is going to go all the way down towards the axon terminal. So once this sodium ion gated channel is open, we have very quick rush in of the sodium ions. Now you have these sodium ions all happening, existing within the cytosol of the neuron. And we are at this point of the graph where we're climbing up to positive 30 millivolts. And once we are at positive 30 millivolts, that's the peak right here of our action potential graph. And sodium channel now is closed because you're at this positive 30 millivolts. And instead, your potassium channel will now open, allowing potassium ions to move out of the cell. And now you have potassium in the exterior part of the cell, and you will decrease this membrane potential back downwards because you're allowing more positive charge ion to move out of the cell. So then what happens is we will get to negative 70 and even past negative 70 to negative 90 in this temporary hyperpolarization state because your potassium channel needs time to close. It just begins closing at negative 70 millivolts. So by the time it's all closed, it's already overshot in terms of membrane potential to get to that negative 90. And what happens is now, so the rest of that goes out to give you that negative 90. And we're at this point in the action potential graph. And we're in the refractory period where the resting membrane potential would just slowly climb back upwards towards resting membrane potential once both sodium and potassium gated channels are both closed, completely closed. The natural permeability of the plasma membrane, of the neuron's membrane to these ions will reset the membrane potential back to negative 70. And also your sodium potassium pump will contribute to that by ejecting 3 sodium ions for every 2 potassium ions that come through. Make sure you know what sodium potassium exchange pump does, how many ions and what ion it moves out of the cell, and how many ions and which ion it moves into the cell, and the fact that it requires ATP. It's a pump, which means it's an active transporter, okay, that requires ATP to do its job. Okay, All right, and then you should be back at membrane resting potential that will set those ions back to their original position, because you see at this end of the action potential here, you see potassium is now mostly on the outside and sodium now is mostly on the inside. So this pump has to reset the ions back to their original position with more sodium on the outside and more potassium on the inside, and the membrane potential back to -70, and then you're ready for another round of action potential. Okay, so remember, we talked about how some of the neurons have myelin sheaths around their axons. So we will now discuss their importance in application to an action potential here. So in a neuron without myelin, there are many sodium ion channels down the entire length of its axon. And in order for the action potential to progress, all of them needs to be open one by one. Okay, so here's a sodium channel, here's a sodium channel, here's a sodium ion channel. All of them has to be open one by one. And this is referred to as continuous propagation of an action potential. So for example, if this is an unmyelinated neuron with its axon and then its axon terminal there, you have almost if one of those dashed line represents a sodium ion channel, that's about how many sodium ion channel you're going to have to open in order for the action potential to be propagated all the way down to the axon terminal. Now, in a neuron, on the other hand, if a neuron has what we call myelin sheath, we call it as myelinated neuron. These are those with myelin sheath surrounding areas of the axon that creates areas of internodes and areas of a node here. Okay, so what happens is there will be less amount of sodium ion channels to open in myelinated neuron, because those areas that are covered by myelin will not have sodium ion channels. They're covered by phospholipids and proteins, so there's no channels going through those membrane in those areas, in these yellow myelinated area. So after opening one sodium channel here, for example, this is where it first started, the local current will then hop over to the next sodium channel here, okay? And it hops over the entire internode region that's formed by the myelin sheath to the next available voltage gated ion channel here. Okay, and then it hops over to the next ion gated channel here, so on and so forth. It keeps hopping and hopping. Okay, so in a sense, instead of having to open all of these sodium channels, we have these areas where it's covered up by myelin. Okay, so you have less amount of sodium channels to open when you have a myelinated neuron. Okay, this is called saltatory propagation, and this results in an action potential that happens much more rapidly than a continuous propagation, where you have to open countless amounts of sodium ion channels before you get to the end. So here's a good demo video that shows the difference between continuous versus saltatory propagations. Imagine that each of these dominoes here represents a sodium ion channel, for example. Okay? And what you have here in comparison is the one here with the purple paper is myelin. Okay? So this here represents a myelinated neuron, and this here represents an unmyelinated neuron. Okay, unmyelinated, excuse me. Okay, so what happens is this represents the numerous amount of dominoes, of sodium channels this unmyelinated neuron has, whereas here you can skip and hop over these areas of myelin, and you will see which one results in a faster reaction. And you can see here, the myelinated neuron is already done. And we're still waiting for a couple seconds for that unmyelinated neuron to continue and finish opening up its sodium channel. So this is a direct illustration of why an unmyelinated neuron is conducts a lot slower than a myelinated neuron in terms of an action potential. Okay. So now what happens at the axon terminal once an action potential has arrived at the location of the neuron? Okay, so we got to finish the story here. We just went through the action potential part. So we kind of had a glimpse of it when we talked about the neuromuscular junction. But instead of just talking about an impulse propagation from a neuron to a muscle fiber, now we're talking about from one neuron to the next neuron. Okay, and for example, from a sensory neuron to an interneuron. So that's what we talked about, is that they got to synapse with each other in order to allow that electrical signal to go through sensory, interneuron, central nervous system, and then maybe to another interneuron, and then back out in your motor neuron. So at the end of one neuron, the axon terminal would synapse on another neuron's dendrites or cell body. And synapse is what we call a specialized site where the neuron communicates with another cell, whether it is a neuron or a muscle fiber. And their information always travels from what we call this as the presynaptic neuron to the one receiving the signal as postsynaptic neuron. Okay? And a chemical synapse is when one neuron sends chemical signal to another cell, for example, a second neuron, while it travels, those chemical signal has to travel through that little gap we talked about in neuromuscular junction called synaptic cleft. So you still have this existence of the synaptic cleft there, but now it's just between neuron number 1 and neuron number 2, or presynaptic neuron and postsynaptic neuron, that separates the two neuron cells. Now each axon terminal actually contain a gazillion, lots of these mitochondria, and thousands of these green balls here, which is those vesicles, synaptic vesicles we talked about. And they're filled with neurotransmitter molecules ready to be released into the synaptic cleft. Now, what happens is, remember we talked about acetylcholines are either broken down or dissolve away. So when it is broken down, the axon terminal can reabsorb those broken products and either reassemble it here or actually bring it all the way back to the cell body for it to be resynthesized again back into a normal functional neurotransmitter, so they can refill those synaptic vesicles, always ready for that action potential to arrive and release the action potential into the synaptic cleft. So synapses that release acetylcholine, remember there's a lot of neurotransmitter. They don't necessarily all have to release acetylcholine. They can release something else. But those that release acetylcholine is called cholinergic synapses. There's names for different neurotransmitter synapses, but acetylcholine is the most widespread of all the different types of synapses you can have in the body, because they always form neuromuscular junction with skeletal muscle fibers, and we have so much skeletal muscle fibers all over our body that needs to be controlled by a motor neuron. So some cholinergic synapses also synapse with the central nervous system, and some within neuron-to-neuron synapses within the peripheral nervous system as well. Okay. Now, what can delay the propagation of an electrical impulse between two neurons is the timing between the arrival of an action potential at the axon terminal and the effect on the postsynaptic membrane, which is we're going to talk about a third ion that's involved in this last step of the story, which is calcium. So calcium was we talked about being needed in the membrane, in the muscle fiber for muscle contraction by binding the troponin. But here, between synapses at the axon terminal, calcium is also very much needed for activating of neurotransmitter release. I'll talk about this in the diagram in the next slide. And hence, because we have to wait for calcium to come into the axon terminal to activate the excitatosis of neurotransmitter, the more synapses there are, the more we have to wait around for these neurotransmitters to be released in the synaptic cleft. And hence, the fewer synapses the route going from a sensory neuron to a motor neuron has, the faster the response, because more synapses means more time accumulated to wait for that influx of calcium ion and neurotransmitter release. So fewer synapses, faster responses. Every time there's a synapse going on between an interneuron and whether it be sensory or motor neuron, you have to wait for that, release of neurotransmitter and influx of calcium ions to happen before the propagation of the signal occurs. So that's why more complex things that involves more interneurons, it's a little slower compared to very quick reflexes. And some reflexes go from one single sensory neuron to the spinal cord, doesn't even need to go to the brain, and comes right back out in a motor neuron very, very quickly. And that's why sometimes we act in very, very fast mode, because it is not requiring a lot of synapses with interneurons. Okay, so here's what needs to happen when action potential arrives at the axon terminal of the presynaptic neuron. Now, presynaptic meaning, as I've talked about, that's the one that needs to release the neurotransmitter so the message can be relayed to the next neuron, which is the postsynaptic neuron. Okay, the one receiving the message is postsynaptic neuron. So the action potential opens the voltage-gated calcium channels here. So here's this action potential coming down towards the axon terminal. Then it reaches the calcium voltage gated channel, activates it, allow calcium ions to flow through. Calcium ion then trigger exocytosis of acetylcholine from the synaptic vesicles. And once acetylcholine can travel into the synaptic cleft here, it binds to the postsynaptic membrane chemical gated sodium channel activated by acetylcholine's binding and allows sodium ions to flow into the cell, therefore propagating that action potential further down in the postsynaptic neuron. Okay, so that's how we go from a presynaptic neuron to activating that same signal in a postsynaptic neuron by the action potential arriving at axon terminal, causing calcium ion channels to be activated, causing exocytosis of acetylcholine, which will bind to the postsynaptic neuron, or in neuromuscular junctions case, this was the muscle fiber. Okay, and because we talked about the what is graded potential, the more acetylcholine there is that's released in the synaptic cleft, the more ion channel that will open. Okay, or the longer it will stay open, the longer you have acetylcholine remaining in there. And that's why sometimes when we don't want the stimulus to keep going, and when the stimulus is done, okay, the acetylcholine can be removed from the synaptic cleft by your acetylcholinesterase that can break down the acetylcholine, and then all these broken parts can be reabsorbed back in the axon terminal and reform back into acetylcholine. Okay. And as I've mentioned before, there are lots of different neurotransmitters in the nervous system. It's not just acetylcholine. And here's a list of common classes of neurotransmitter and what they do. So for example, there's norepinephrine, which is a widely distributed neurotransmitter within the brain and the autonomic nervous system. So you will see norepinephrine's action when we talk about AMP2. we'll talk about the difference between release of epinephrine versus norepinephrine, or with acetylcholine. Okay, so norepinephrine typically is an excitatory depolarizing effect on the postsynaptic membrane, meaning it does lead to an action potential. Dopamine, on the other hand, is a central nervous system neurotransmitter that can act in two ways. It can act as an inhibitory effect that allows for precise control of movements, because if you have overstimulation of neurons and no inhibitory effect at the right moment, you won't have that smooth movement of a muscle. Okay, so this is what happens in people with Parkinson's disease, is that they don't have enough dopamine in their synaptic cleft in those skeletal muscles, and it leads to rigidity and stiffness because you don't have that inhibitory effect of that to produce that smooth movement a normal person would have. Excitatory effect is the other thing dopamine can do, and this is what's responsible for that high feeling one gets, that one, for example, drug addicts experience, with cocaine users. And because what happens with them is, and that's why they call drug dope, okay, because it is from release overstimulation of dopamine and keeping those dopamine within the synaptic cleft to give you that happiness feeling. And we're supposed to get dopamine a little bit to make us feel happy about ourselves, but we can't have prolonged periods of dopamine for no reason at all. And that's what that drug does, is to keep dopamine from breaking down within the synaptic cleft. Okay, and serotonin, everybody has heard of this. This is another central nervous system neurotransmitter, and if you have inadequate amount of serotonin, it can affect a person's attention and emotional state. So people that have depression problems tend to need to take drugs that keep serotonin longer within the synaptic cleft, so we can manage our feelings and emotions. And that's what Prozac, Paxil, and Zoloft are commonly used for as antidepressant drugs that inhibit reabsorption of serotonin from the synaptic cleft. GABA, gamma aminobutyric acid, is generally inhibitory, okay? And 20% of synapses in the brain releases GABA, but again, function of GABA is still incompletely understood. But what the best hypothesis researchers can come up with is it appears to reduce anxiety when you have enough GABA around. Other than neurotransmitter, there's another group of chemical called neuromodulators. What neuromodulators are, they are neuropeptides that can alter how much of the neurotransmitter is being released by the presynaptic neuron into the synaptic cleft. Okay? For example, opioids is an example of a neuromodulator. And for example, drugs such as opium or morphine, we all know they're for relieving pain. You don't feel that sensation of pain as much when you have morphine injected into the body, is because morphine will inhibit the release of another neurotransmitter called substance P, which is mainly for relaying pain sensation. So if you block substance P from being released into the synaptic cleft, then you don't feel the pain, because none of that sensory information is being relayed to the central nervous system. Okay, so and as we have seen with dopamine, neurotransmitter can have an excitatory effect or an inhibitory effect. What do we mean by that? It all depends on what ion channels the neurotransmitter will trigger to open. If the neurotransmitter triggers opening of a sodium ion channel, which is a positive ion, then it will raise the membrane potential from negative to be more positive and lead to activation of an action potential. This is called EPSP, or excitatory postsynaptic potential. Okay, so for example, glutamate here, other than acetylcholine, there's other things too. So glutamate here activates the chemical gated ion sodium ion channel and allows sodium ions to influx into the postsynaptic neuron, raising the membrane potential up. Okay, so that's what we call and what we have seen as an EPSP. Okay, it excites the neuron, activate an action potential downwards. Okay. On the other hand, if the neurotransmitter opens, for example, a chloride ion channel instead, it's a negative charge ion for chloride. So influx of chloride ions here will only bring down the membrane potential even further to be more negative than it originally was. And it is now harder to get to that threshold and undergo depolarization. For example, originally, let's say this was -70 to begin with as resting membrane potential. Now, because GABA is inhibitory neurotransmitter, it activates your chloride ion channel instead. Inflow comes chloride ion. It's negative charge, so it's only going to make this membrane even more negative. Let's say -85 or something, or 90. And now you have a greater membrane potential to get to reach that positive -60 to activate the neuron. Okay, so you have even more of a travel distance before you can reach an excitatory EPSP. So that's why we refer to this as IPSP, or inhibitory postsynaptic potential. Okay. So another way that an IPSP can happen is if the neurotransmitter open chemically gated potassium channel, and there are those too. We've just been talking about voltage gated potassium channel down the length of the axon for repolarization, but at the dendrites, for example, or cell body of a neuron, for example, there can be chemically gated potassium channel where a neurotransmitter can bind to a potassium channel, open the potassium channel, cause potassium channel to flow out of the cell. Okay, and in that sense, you have potassium channel now going outwards from the cell, and that can also result in a more negative membrane potential to start with. Okay, and that's why that is also another way IPSP can happen. And that's what we mean by here, what I was talking about earlier. Okay, all right, so just as I have talked about graded potential, one individual EPSP has only a small effect on membrane potential. So they must be combined through summation that integrates all of the graded potential before you can activate the trigger for an action potential. So there are two types of EPSP summation: temporal versus spatial summation. In temporal summation, the stimulus will arrive successively in rapid succession at a single synapse location on the neuron, okay, whether it be a dendrite or the cell body, but it continuously arrives at that one location of the neuron, okay. First stimulus arrives, second stimulus arrive at that same location via that same axon terminal, and eventually it reaches threshold, activate an action potential down the length of the axon. Now, every time an action potential arrives, groups of vesicles of acetylcholine is released. And I talked about how if there's more acetylcholine, more sodium ion channels are open, more depolarization, which will lead to threshold. So that's what this whole temporal summation is about. More acetylcholine coming through, being released in the synaptic cleft because you have continuous stimulus coming through from the presynaptic neuron. Okay, now spatial summation is a second type of EPSP summation where two stimulus are arriving at different locations on the postsynaptic neuron simultaneously, resulting in that same cumulative effect on the membrane potential that will activate your action potential onwards in the axon. So it's just whether you're having many stimulus coming through at that one location location or two location from two different axon terminals arriving at this one neuron, and they're stimulating that neuron at the same time until it reaches that action potential threshold. Okay? And we will end this chapter with a type of neural disease that is specifically acting on neurons called demyelinating disease. Okay? Demyelination is a progressive destruction of myelin sheaths in both the central nervous system and peripheral nervous system, which can lead to, of course, loss in sensation. You can't feel anything, and loss of motor control because your motor neurons are not working effectively. Okay, because now you have delayed action potential when you don't have myelin. Remember, the purpose of myelin is to speed up the action potential so you can receive those sensory information quick enough and you can act in quick enough. But now you don't have myelin, and the action potential is taking forever to get to your central nervous system. So your hand can be on that burning stove for the longest time and yet is not getting to your spinal cord because of not having enough myelin. So your hand remains on the stove until it burns to a 3rd-degree burn. Okay, so that's what we mean by demyelination. It slows down all the action potential. Now, there can be a number of underlying causes for demyelinating disease. For example, chronic exposure to heavy metal ions such as arsenic, lead, and mercury can lead to demyelination. I'm not going to talk about the mechanism of that, but it can cause that. Diphtheria is a bacterial disease that where the bacteria specifically damages Schwann cells, and in the sense, if they destroy Schwann cells, they will destroy myelin sheaths along the way, and that usually happens within just the peripheral nervous system, which can lead to fatal paralysis as well, kind of like rabies. But instead of infecting our neurons through retrograde, now we're talking about destroying our myelin that's supposed to speed up our action potentials. Multiple sclerosis is an autoimmune disease where somehow your immune system starts attacking myelin out of nowhere all of a sudden. Okay, so usually affect axons in the optic nerve, the brain, and spinal cord. Okay, mainly your central nervous system, really, and that your immune system just starts demyelinating your axons. And for these people, healing can produce multiple scars because we're talking about central nervous system here, and we already talked about damages within the central nervous system does not get repaired but produce scar tissues by astrocytes. So you would actually see these on an MRI scan, for example, of the brain. You will see these lesions in the brain that result from those scars healing after a myelin attack from your immune cells. And depends what location these lesions are, you can lead to partial loss of vision, problem with speech, balance, motor coordination, bowel urinary control. It depends where in the brain is affecting. And because it's an autoimmune problem, this can actually go through cycles of attack and recovery, until the entire brain loses its function. Okay, so it's a pretty scary disease when we talk about multiple sclerosis. All right, and that's all I have for you for chapter 12 on the nervous tissue. Let's go through practice questions and we'll be done. So there are two functional divisions of the peripheral nervous system. Okay, those are the afferent and efferent divisions. What are their respective functions? Afferent, arrival, remember? So arrival of a sensory input to the central nervous system. Efferent for exit of motor commands to either your effector tissue such as muscles or glands. The synapse between a neuron and a muscle cell is called a neuromuscular junction, as we talked about in the previous chapter. Since neurons lack what? They cannot undergo what? They lack centrioles, so they can't do cell division. The functional classification of neurons that are responsible for integrating sensory information with motor output are called what? And these neurons are where are they located? Interneurons that are sitting between sensory and motor neurons. And they're exclusively within the central nervous system, either in the brain or the spinal cord. Which of the central nervous system neuroglial cell function as phagocytic cells? Microglia. Okay. Neuroglia of the peripheral nervous system include just two types of cells: satellite cells and Schwann cells. Both starts with S. Okay, so that's an easy way to remember. Which type of neuroglia would be found lining the hollow regions of the central nervous system? Ependymal cells. Remember, those are the ones, neuroglia cells that are responsible for producing cerebrospinal fluid and circulating them around the central nervous system. The effects of neurotransmitter on the postsynaptic membrane depend on the type of receptor. Okay, different receptor can have different effects. That's what the whole thing was about when we talk about EPSP versus an IPSP. Depending whether the receptor is a sodium gated channel or a chloride gated channel, you can have either inhibitory or excitatory effect. What effect would blocking voltage-gated calcium channel at a cholinergic synapse have on the synaptic communication? This is for ACH to release. So now you're blocking calcium channels, which means no release of neurotransmitter. The whole communication would just cease. It just stops. What structure of a neuron is capable of propagating electrical impulse? Axon. Okay. Dendrite is for receiving things. Cell body also. Okay, perikaryon is just a cytoplasm of the cell body. Enhancing effects of the neurotransmitter. What has allowed pharmaceutical companies to produce medication to decrease anxiety? So GABA, we said, has something to do with anxiety. Effectors include which of the following? All of these, okay? They can be anything. A person you know has experienced sleep problems and prolonged depression. Which neurotransmitter might be in short supply? Depression. Emotion control, so serotonin. Excitatory neurotransmitter cause what? And inhibitory neurotransmitter causes what? EPSP causes depolarization. IPSP causes hyperpolarization that makes the membrane more negative than it should, which makes it harder to start an action potential. Okay, this one forget about because I skipped over the diameter part. This chapter is long enough. We don't need to add any more details. So I skipped over the diameter of the axon. Oh, wait, this is not about diameter. This is about myelinated versus non-myelinated, okay? So axon A is much slower, much faster because 50 meters per second versus axon B propagates at 1 meter per second. So axon A is a lot faster. So what is the structural difference between these axons? So this is very different speed, and axon A is myelinated. So that's why it's a lot faster, okay? Okay, which of the choices below correctly list the steps of an action potential? So you go from depolarization to threshold, activation of sodium channels, which leads to depolarization, all the way up to positive 30, and activation of sodium channels and activation of potassium channels, return it back to normal permeability. Okay, so that whole graph I drew for you would be very helpful in remembering the steps of an action potential. Okay, note that if you read chapter 12 in the textbook, I've already skipped some parts of chapter 12. It's just long and a lot of information. Which of the following statements are true with regard to saltatory propagation? Both A and B are correct. They involve myelinated areas which accelerate propagation, and only nodes would be where you have depolarization stimulus, where the sodium channels are. In what ways are temporal and spatial summation the same? They both are causing individual EPSC to combine. Summation of those graded potential that's going to activate an action potential. Okay, and that's all I have for you for this chapter.